So thank you all, and please have a seat. I want to welcome all of you to the Harvard Medical School PhD Hooding for 2024. Welcome graduates, welcome families, faculty, program leaders, program administrators. Special welcome to Dean Dench and the entire Ken Griffin GSAS school leadership who make such an effort to come over here for this, which we really appreciate. I'm Roz Siegel, the Dean for Graduate Education at HMS, and the co-director of Harvard Integrated Life Sciences, together with my great friend and colleague, Elena Kramer. It is such a pleasure to be able to come together and celebrate our wonderful and talented colleagues, the new graduates. Each one of the graduates has carried out important and groundbreaking research. We now have the opportunity to cheer them as they move on to new endeavors in life sciences, and we can clap again. So this program is centered on the graduates, so I just am leading the proceedings. So I would now like to introduce our student speaker, the newly minted Dr. Allie Stanton. Allie first became interested in virology as a high school student who came down with mono. Instead of binging Netflix, she delved into studies of Epstein-Barr virus and its involvement in mono as well as in cancer. She therefore went on to attend college at MIT, where she majored in biology and mathematics. At MIT, she dug into multiple aspects of virology, including flu vaccines and evolutionary studies of viruses across species. As a Harvard virology PhD student, she's worked in the laboratory of Dr. Pardis Sabeti. Her work has addressed both practical approaches for developing viral tools to be used in research and in the clinic, and more conceptual studies aimed at understanding the mechanisms that regulate the translation and production of viral proteins. In addition to her beautiful work in the lab, Allie has been very involved in the Graduate Student Union. She has been a dedicated teaching fellow and has been at the center of the virology community. Her work and her scholarship has been recognized by the Herschel Smith Fellowship. After graduation, Allie plans to continue to pursue her passion for virology as a postdoc, and we hope that this will be in our community at HMS. I look forward to hearing her talk today.
Thank you for the very kind introduction, Roz. And welcome faculty and staff, friends and family members, and of course, my fellow graduates. It is truly an honor to be addressing you all on this occasion. I can speak for all of us when I say that the many long days and sometimes even long nights over the course of 4, 5, or 6, or 7, or 8 years have finally been realized as we now celebrate successful dissertation defenses and receive our long-awaited doctoral degrees. Personally, I could have gone without the ceremony, since the many trials of my own PhD were instantly vindicated when I went to renew my driver's license at the RMV and instructed them to change my title from Ms. to Doctor. At this very moment, I'm sure many of your family members are biding their time until my speech and this ceremony are over, and it will be their turn to regale you with stories of your early forays into scientific inquiry. All of this is a prelude to the real celebration, in which you will be told of all the dinosaur names you memorized, bugs you collected, and poorly controlled experiments you performed with various household chemicals. My earliest research projects involved picking scabs off of my elbows and looking at them under a children's microscope. I'm still waiting to hear back from nature on that one. But I really became a scientist because of AIDS. I came out of the closet when I was 13, and it didn't escape my notice at the time that there were very few queer adults around me that I could look to. And a big part of why that was the case was because many of the people a generation or two older than me died of AIDS in the 1980s and 1990s. Not too long ago, an HIV diagnosis meant an early and uncomfortable death, with your remaining time spent managing the debilitating side effects of perhaps a dozen different medications that did little to slow the course of the disease. A few decades later, with access to good medical care, a person with HIV can take just 1 or 2 pills a day and live a normal lifespan without ever developing symptoms of AIDS or risking transmission to anyone else. Painstaking advancements made by thousands and thousands of dedicated basic science researchers is bringing AIDS to heal, just as our predecessors tamed diseases like smallpox, polio, and diphtheria, to the point that they are easier to find as answers to trivia questions than as horrors plaguing children's hospitals. Because of the kind of people like you, people who go to graduate school and get PhDs and do scientific research, I have many wonderful queer friends who are very much alive, and we spend the warm, sunny afternoons of our 20s and 30s attending potlucks and open mic nights instead of funerals. So the engineers and physicists can have their moon landing because our greatest and most benevolent achievements as a species have in fact been gifted to humankind by practitioners of the life sciences. And as I imagine is the case for many of you, I became a biologist not just because of a sense of curiosity, but because I wanted to contribute to this legacy. Almost nobody is capable of changing the whole world as a single person, and like most things, science is a cumulative effort. But I think all of us are here, at least in part, because we wanted to add a small crumb to the totality of knowledge about the natural world, to increase our understanding of the biological systems underlying health and disease, or in other words, to do good. But invariably, we've had moments that challenged us in our graduate careers. Failed experiments, negative reviewer comments, rejected papers and grant applications, and the endless march of deadlines have tested our determination. There have probably been times where you began to question why you are here, and if it is even possible to do something that matters. Earlier, I may have implied that because of the efforts of biologists, AIDS is more or less a solved problem. This is not correct. For many people, AIDS is a shadow of its former self, a chronic condition like diabetes that can be managed with the intention of a capable primary care physician. But in 2022, an estimated 630,000 people died of AIDS. Despite the objectively enormous strides we have made in understanding and treating HIV infections, despite the work of many thousands of dedicated and creative scientists, annual AIDS-related deaths are at similar levels as they were in the early 1990s. The world is sicker than it should be, even in spite of the impressive collective research output of this room. The last few years have made this clearer than ever. The COVID-19 pandemic not only impacted each of us individually, but transformed the public's relationship with the field of biology. In March of 2020, I was in my home, considering the implications it had for my sense of purpose that I was a virologist living through a viral pandemic, and my primary activity was watching reruns of The Bachelorette with my roommates. 2 years later, we watched as the Supreme Court overturned Roe v. Wade, and reproductive care has become out of reach for many. We're now inundated with images of war, the slaughter of civilians, and the destruction of hospitals in Ukraine and in Gaza. While we can sometimes celebrate a new finding at the bench that translates to a better outcome for people who are sick, receiving any kind of medical care often comes at the cost of a lifetime of medical debt. The correlation between research progress and the health and welfare of humankind is only a loose one. Last year, I had the privilege of teaching Life Sciences 1B, an introductory genetics lab class for undergraduates at Harvard College. This was an excellent forum for me to demonstrate my skills as a biologist, specifically the skill of Googling things like, how many chromosomes do humans have? And what are the steps of meiosis? It was also an opportunity to witness a love for the natural world blossom in the minds of some of my students. We had one unit that involved a lab where students had to use a microscope to observe transgenic C. elegans roundworms that had been modified so certain cell types would glow green when you shine a light on them. I had one student who had experienced a personal tragedy that semester. She stayed behind for half an hour after finishing the lab to keep looking at those worms under the microscope. And for the first time in weeks, she left my classroom with a smile on her face. In moments like that, I am reminded how lucky we are to be able to make a job out of appreciating the sublime beauty of biology. I've also had the great honor to be a shop steward for the Harvard Graduate Students Union, which is our labor union here and a local of United Auto Workers. This was an opportunity not just to serve my fellow scientists, but also to connect what is inherently a community effort of science to labor struggle and to social struggle. When all scientists, including trainee scientists, recognize their agency in the world and take hold of their collective power, we have the ability to do incredible good and bring us closer to a new era where democracy and humanitarian concerns are at the forefront of scientific endeavor. Even in a chaotic world, science still matters, both for its potential to increase human health and for the love of knowledge itself. But in order for it to matter as much as it should, we have to recognize that our job does not end at the threshold of the lab. Science is not inherently good, and without our conscientious intervention, it can be used, abused, or neglected. So if we want to see the fruits of our labor, we cannot just be scientific investigators, but also the communicators, advocates, and champions of science as a tool for bringing a better world into being. The last few years have seen an explosion of citizen science initiatives, where the broader public can engage with and contribute to research, every citizen a scientist. We should also consider the importance of the converse, that every scientist is also a citizen and an activist. The pursuit of science and the pursuit of truth and justice are intrinsically linked. But no one else is better positioned than we are to help guide society towards a wise and humanitarian utilization of science for the collective benefit of humankind. All it takes is for us to step outside into the wider world. And with that, I'm really looking forward to seeing what kind of difference you all will make. Thank you.
I want to thank Allie for that really inspirational talk that really speaks to what we're trying to do here. It is now my great pleasure to introduce Dr. Wendy Garrett, who will read out the names, advisors, and dissertation titles of each graduate. I'm so happy that she is doing this. Dr. Garrett is the Irene Heinz Given Professor of Immunology and Infectious Diseases in the Department of Immunology and Infectious Disease and of Molecular Metabolism at the Harvard Chan School of Public Health. She's a professor of medicine at Harvard Medical School and the Associate Director of the PhD program in immunology. Wendy?
Biological and Biomedical Sciences, Nuran Abdel Fattah, Advisor, Stephen Elledge. T-Switch, a specificity-based engineering platform for the development of T-cell therapeutics. Beatrice Augusti, Advisor, Kevin Haggis, in vivo tissue-specific multiomics analysis of EGFR-stimulated EGFR signaling. Courtney Benoit, Advisor, Tracy Young Pierce, Regulation of Neurodevelopment and Neural Maturation by the Transcription Factor, PU3F2. Alexander Andrew Bigger Allen, Advisor, Rosalind Adam, Assessment of the Impact of the bromodomain inhibition on the actin cytoskeleton and contraction. Stephen Charles Berger. Advisor, Stephen McCarroll, X inactivation, mosaicism, and the effects of X-linked mutations in the human brain. Ana Carla Severa Diaz, Advisor, Jeff Dvorin, building and maintaining the ultrastructure of Plasmodium falciparum parasites during the asexual blood stage. Ziki Chen, Advisor, Ulrich von Andrian, modulation of T-cell trafficking of the small and large intestine mucosa. Noah Connolly, Advisor, Shamol Sunyev, the functional effects and fitness consequences of GWAS loci. Benedetto Di Ciaccio, Advisor, Cornelia Polyak, modulators of sensitivity to KDM5 inhibition in basal breast cancer. Kelsey Alexandria Everett, Advisor, David Liu, precision genome editing strategies to treat sickle cell disease and CDKL5 deficiency disorder. Elaine Fivenson, Advisor, Thomas Bernhardt, the regulation of outer membrane biogenesis and its role in cell morphology. Stanley Gill, advisors, Kevin Egan and Lee Rubin, building in vitro human iPSC-derived motor neuron systems to explore cell autonomous mechanisms and modifiers of TDP-43 perturbation-associated neurotoxicity. Alyssa Goss, advisor, Anna Greca, TDMEDs are differential regulators of mutant membrane proteins. Sarah Hakim, advisor, Clifford Wolf, neuroimmune mechanisms of diabetic neuropathy. Ayana Lachey Henderson, advisor, Monica Kolayakova, assessing how benzobutyl phthalate exposure impairs the germline using C. elegans. Samantha Elena Hoffman, advisor, Eliezer Van Allen, multiomic insights into gastroenteropancreatic neuroendocrine tumor biology. Reina Iwasi, advisor, Philip Cole, semi-synthetic approach to explore the N-terminal regulation of tumor suppressor PTEN. Nina Jane, advisor, George Church, factors affecting AAV production, mutagenesis of AAV2 rep and AAV transgenes. Dawn Jung, advisor, Mariella Philbin, multidimensional profiling of tumor cell-specific heterogeneity reveals cell lineage-specific functions in supratentorial ependymomas. Liam Project Kelly, advisor, Marcia Haggis, multiomics interrogation of metabolic stress and cancer. Sharon Heejin Kim, advisor, Vamsi Mutha, the Gordian knot of oxygen and iron. Valentino Lagomarsino, advisor, Meenakshi Rao, microbial metabolism of host androgens regulates enteric nervous cell system function. Yang Lim, advisor, Johannes Walter, Donson, a novel metazoan DNA replication initiation factor. Yue Jane Liu, advisor, Sarah Fortune, elucidation of an essential genetic pathway under antibiotic selection in Mycobacterium tuberculosis. Chancia C. Ma, advisor, Christopher Welsh, genomic studies in chronic traumatic encephalopathy from external traumas to genetic alterations. Jorge Diego Martin Rufino, advisors, Eric Landra and Vijay Sankaran, decoding influences of genetic variation on gene regulation in blood and immune cell development. Michael Mazzola, advisor. David Scadden, distinct translation initiation strategies during chemopoietic differentiation. Claudia Abrantes Mimoso, advisor, Karen Edelman, defining the crosstalk between the transcription and splicing machineries. Alexander Morano, advisor, Jeffrey DeVoren, structure and function of the basal complex, the unique mechanism of plasmodium cytokinesis. Christopher Nardone, advisor, Stephen Elledge, the control of transcription factor degradation by ubiquitination-dependent and independent mechanisms. Adria June Navarro, advisor, Alex Toker, the glycosyltransferase alpha-1 3-minosyl-transferase, ALG3, in an AKT substrate that regulates protein and glycosylation in breast cancer cells. Smriti Pandey, advisor, David Liu, enhancing precision genome editing agents through direct evolution and protein engineering. Alexandra Panoff, Advisor Wade Harper. The roles of proteasome-associated factors RPM13, UCHL5, and UBE3C in basal unfolded and aggregated protein turnover. John Luis Police III, Advisor Matthew Meyerson, amplification and dosage in the oncogenic regulation of the NKX2-1 lineage transcription factor. Alexander Radakovich, Advisor Jack Sostak, primordial role for RNA amino isolation in the assembly of structured and functional chimeric RNAs. Anita Reddy, Advisor Edward Chuchani, examining the role of succinate signaling in tissue remodeling. Alexander Anthony Souza, Advisor David Liu, enhancement and application of prime editing to correct pathogenic mutations in vitro and in vivo. Lauren Sullivan, Advisor Angela DePace, dynamic expression of pleiotropic developmental genes during Drosophila embryogenesis. Tristan Tay, Advisor Jason Buenrostro, engineering the epigenetic regulatory networks of T-cell exhaustion and embryonic stem cell differentiation with transcription factor perturbations. Do my best. Aseda Tena, Advisor Frederick Alt, mechanisms of recurrent DNA break clusters, RDCs, generation in mouse and human cells. Tommy Tullius, Advisor Sterling Churchman, FiberSeq reveals the single molecule architecture of nuclear transcription in the mitochondrial genome. Yilan Wang, Advisor E. Alice Lee, discovery of ultra-low signal variants to study human evolution and epilepsy. Irene Garling Wong, Advisor Carla Kim, tissue-wide responses to airway injury. Shane Zhou, Advisor Yang Shi, discovery of cancer vulnerabilities in gene expression regulation pathway. Samuel Evan Zimmerman, advisors Alexander Kostic and Chirag Patel, the ecological and temporal genetic architecture of the microbiome in health and disease. Biomedical Informatics, Richard Chen, Advisor Faisal Mahmood, Multimodal and Context-Aware Computational Pathology. Tang Gao, Peter Park, and Peter Charchenko, Somatic Genome Evolution in Cancer and Aging from the Lens of Transcription. Reihun Zhang, Advisor Martha Bullock, The Effects of Trait Association Variation on Transcription Factor Binding and Other Regulatory Phenotypes. Laurie Rumker, Advisor Sumyure Chowdhury, Modeling Inter-Individual Variation in Single-Cell Datasets to Detect Cell State Abundance Associations to Clinical Features and Genetic Variants. Luke William Sagers, Advisor Arjun Manray, Augmenting Medical Imaging Classifiers with Synthetic Data Across Populations. Saeed Rakeen Ahmed, Advisors Jair Shri, Kalpathy Kramer, and Bruce Rosen, Generating Clinical Translational AI Models for Cancer Diagnostics. Daniel Barabasi, Advisor Florian Engert, A Hobbit's Guide to Neurodevelopment, An Unexpected Journey into the Innate. Immunology. Katojit Chowdhury, Advisors Christophe Benoit and Diane Mathis, Decoding Regulatory T-Cell Transcription Factor Networks from Identity to Diversity. Tashika Jawick-Rema, Advisors Diane Mathis and Christophe Benoit, Lymph node stromal cell responses to perinatal T-cell waves. Nia Millard, Advisor Sumyure Chowdhury, Methods for the Design and Analysis of Disease-Oriented Multisample Single-Cell Studies. Anusha Nathan, Advisor Gaurav Gaya, Rational Design of CD8 positive T-cell vaccines for SARS-CoV-2. Alexandra Anna Prohera, Advisor Meenakshi Rao, Enteric glial functions and gastrointestinal homeostasis. Thomas Su, Advisor Shiv Pillay, Surface display of proteins on bone marrow-derived dendritic cells induces humoral immune responses and anaphylaxis. Program in Neuroscience, Diego Etione Alderando, Advisor Ben Sialveski, Modeling the Neural Control of Movement with a Virtual Rodent. David Harrison Brand, Advisor Sandeep Datta, Gene Expression Programs in the Nose. Wendy Fung, Advisor Maxwell Heyman, Patterning of the Apical Extracellular Matrix at Sense Organs. Winthrop Forrest Gillis, Advisor Sandeep Datta, Basal Ganglia Dynamics during Spontaneous Behavior. Alexander Greben, Advisor Michael Greenberg, Chromatin Regulation of Transcriptional Repression in the Developing Brain. Michael Gullidge, Advisor Elena Chartoff, Spontaneous oxycodone withdrawal disrupts sleep, circadian, and electrophysiological dynamics in rats. Alexandra Hartman, Advisor Rachel Wilson, a neuron in the visual system that provides feedback about limb movement. Lily Ha, Advisor Nicholas Bolano, Molecular Tuning of Sea Anemone Stinging. Maya J., Advisor Sandeep Datta, Striatal Dopamine Structures Spontaneous Behavior Across Multiple Timescales. Jenny Lai, Advisors E. Alice Lee and Timothy W. Yu, Functional Genomic Dissection of Human Brain Development, Degeneration, and Dysfunction in Pediatric Neurogenetic Disorders. Lucy Lai, Advisor Samuel Gershman, Policy Compression Acting with Limited Cognitive Resources. Lauren Elizabeth Miner, Advisor Michael Crickmore, Motivational Control of Drosophila Copulation Across Timescales. Julia Kwa Win, Advisor Christopher Harvey, Task-Dependent Mouse Behavioral Dynamics and Navigational Decision Making. Jeff Rhodes, Advisor Wei-Cheng Alan Lee, Grounded Inference via Local Signals and Learned Representations by Organic and Artificial Systems. Elena Westind. Advisor Rachel Wilson, connecting internal representations to behavior in the Drosophila navigation system. Stephanie Arro, Advisor Thomas Quattieri, speech, perception, and auditory attention in noisy and multi-talker environments. Speech and hearing bioscience and technology, Daniel Lowe, Advisor Sadarijat Ghosh, speech and text psychometrics, identifying suicide risk factors with large language models and acoustic networks. Stephen Meisler, Advisor John Gabrielli, White Matter Structural Correlates of Reading Abilities, Disabilities, and Intervention. Systems, Synthetic, and Quantitative Biology, Sabina Jehan Hook. Advisor Jeremy Gunawardena, Graph Theoretic Approaches to Biochemical Reaction Networks. Ethan Mackenzie Jones, Advisor Pamela Silver, Design and Construction of Synthetic Biology Tools for Sustainable Lanthanide Extraction. Natalie Quinones Alvera, Advisor Michael Baim, New Methods to Explore Mobile Genetic Elements in Bacteria. Siddharth Raju, Advisor Pardis Sabeti, Pioneering Genomic Technologies and Computational Tools to Study Deadly Pathogens Endemic to Low-Resource Countries. Virology. Gabriel Dugado, Advisor Dan Baruch, Viral Vectors for Gene Therapy, Cancer, and SARS-CoV-2. Jared Evan Feldman, Advisor Aaron Schmidt, Origins of a Humoral Immune Response Characterizing Virus-Specific Naive B Cells. Rafael Hazel Eka, Advisor Victoria D'Souza, developing a polymerase-driven method to incorporate base modifications and structurally characterizing M6A and colobactin-modified nucleic acids. Daniel Patrick Maurer, Advisor Aaron Schmidt, Viral Escape from Antibodies Targeting Conserved Sites. Ajinkya Patel, Advisor Sagal Kadosh, Disruption of Mammalian SWE, SNF, chromatin remodeler function in human disease.
Crushing it. That's sea bass like the fish.
Okay, that's awesome.
Also your question.
Desmond D. Richmond Bukala, Advisor Philip Kransuch. The phage prohead protease is a key determinant of type 1 sea bass immune activation and evasion. Hembley Genevieve Rivas, Advisor James DiCaprio. BMIB and FOXM1 drive cell division through cooperative regulation of cell cycle genes. Alexandra Copeland Stanton, Advisor Pardis Sibetti. Massively parallel approaches to functional characterization of natural and engineered viruses. Last but not least, Irina Stryopunina, Advisor Flaminia Catarruccia. The role of Anopheles gambiae vitellogenin in nutrient transport, fertility, and Plasmodium falciparum development.
Now, I want to, first of all, say a very great thank you to Wendy, who we can all agree did a fantastic job in so not to be competitive or anything, but I think it says that Harvard's maybe better than Jefferson. If anyone has seen those. So we did manage to read all those words correctly, and Wendy does a great job with it. So I think we should have a round of applause for our fabulous graduates who we've all watched being hooded. It's now my great pleasure to introduce Dr. George Daly, who is the Dean of Harvard Medical School and a professor of biological chemistry and molecular pharmacology, and as you saw from his interactions with the graduates, is also involved directly in training. George?
Thank you very, very much, and good evening. I'm immensely proud of our biomedical doctoral programs, and so I want to just start by acknowledging the phenomenal leadership that we have, our Dean of Graduate Education, Roz Siegel, and all of the distinguished, dedicated faculty members who lead our doctoral programs. But I'm delighted to have the opportunity to offer all of you, our newly minted PhDs, my heartfelt congratulations. Again, you can never have enough. I want to begin, though, by sincerely thanking you for all of your perseverance and your resilience over this past year. One's PhD years are never easy, but with all of the crises around the world and all of the strife on the Harvard and MIT campuses, you've been shouldering a lot. But tonight, our focus is on a celebration of this pivotal moment in your careers. Your hard work has gained you a permanent place in the scientific family tree, and from your perch in whatever branch of science you know best, you're going to have a career that will channel your remarkable curiosities into bold experiments and transformative biomedical change. Indeed, your degrees earned at this particular juncture in scientific history are of immense value to the world. There are so many challenges facing the world: aging population, climate change, just a few. And our greatest hope is for scientific discovery. So for all of you, I have one request as you forge your career in this world of challenge. I ask that you always seek the truth, that you always seek to earn the public's trust, and most importantly, that you endure, no matter what the harsh setbacks. You endure to tackle the great challenges because you and your talents are our greatest hope for the future. So today, I salute you. I thank you for your diligence and your efforts. And Harvard Medical School celebrates you and your accomplishments, and we all look forward to seeing the remarkable things that you will all do in your career. Congratulations. And now I believe Dean Siegel will close the ceremonies.
Thank you, Dean Daly, for your inspiring remarks. I find the hooding such a wonderful ceremony that really connects the past, the present, and the future. And really, you guys are the future. I want to finally thank several people who are not on the stage and have been so instrumental to the education and support of our students. First, I'd like to ask our graduates to join me in giving a round of applause to the family and friends who have supported them throughout their lives, and particularly during their path to the PhD. To the family and friends. I also want to thank the wonderful faculty advisors, many of whom you saw today, and the faculty leaders and the program administrators, key people in all of the PhD programs. I want to thank Nancy Gerard and the team from the Office for Graduate Education who organized today's events and made sure we all tried to follow the arrows. Finally, I would ask the audience to please allow the graduates to leave in a procession first as we all move to a reception to celebrate and honor these graduates. And thank all of you for joining us today. Please continue the celebration on the first and second floors of the building and enjoy the evening. So congratulations, everybody.